Last week came to a nice close. I talked to my professor about my project and it seems fairly promising. Things might take a little longer than I wanted to, but it happens. My professor and I talked about what we need to do to get my project from point A to point B. Point A being now, and point B being transforming the diatoms and actually testing them. We talked about different antibiotics to select for our diatoms. There's two different ones, one with a really crazy name and one's called zeosin. The crazy sounding one is like neuroceothristin, which neither of us can pronounce, is a lot less toxic than zeosin, so we're going to end up using that one. And the cool thing about the plasmid that confers resistance to neuroceothristin is that it is expressed using an FCP gene. The FCP gene is one of several different genes that is used in binding photosynthetic pigments to membranes. And that, of course, is obviously going to be highly expressed because it is really, really important to any photosynthetic organism. So essentially, if this plasmid that has the resistance to neuroceothristin is going to be expressed in the cell because the promoter and terminator are going to be highly expressed. And the nice thing about diatoms, which I said before, is that we can transform them with multiple plasmids. So my diatom cells are going to get two different plasmids. They're going to get the GFP plasmid, which is what I was working on last semester, and they're going to get the resistance plasmid. This way we don't have to jam everything that is needed on one plasmid, which is great because we don't know exactly how well the diatoms are going to express what's on the plasmid, you know? It might start at one promoter and only get so far, and it might not get to the point where it expresses both genes. So by having two different plasmids is kind of more of a fail-proof plan, especially because the resistance plasmid is really going to be expressed well because of its good promoter. And that good promoter is the one where it's working with a photosynthetic pigment, so that's always going to be expressed. Or hopefully, you know. So yeah, I went into the lab the other day to finally start growing my plasmids on the plates that I made. These plates have artificial seawater and a gelling agent, agar, and diatoms to grow on them just fine. So I started with a diatom culture, a liquid culture, and I took it off a small amount and I counted cells. Using a microscope takes anywhere from, I don't know, 20 minutes to maybe an hour and a half, depending on how many cultures you're counting, how many cells you want to count, etc. And using a hemocytometer, which is like a little slide with grids on it, you can estimate how many cells are in a solution. And I plated my diatoms based on how many cells were per milliliter, and hopefully they'll grow. But it's going to take about a week or more for them to start showing up on my plates, and then we'll have to, from there, figure out how many cells we need to plate on a plate